Okay, now let's talk about chapter 15, last chapter, and this is kind of that primordial soup chapter. If how are we going to make primordial soup, right? And so, to kind of introduce the topic, they look at different types of wings and how they evolved from the same ancestral tetrapod limb. So petrosaur wings consisted of a membrane primarily supported by one greatly elongated finger. Bird wings consist of feathers supported by an elongated forearm and modified wrist and hand bones. And bat wings consist of a membrane supported by arm bones and four very elongated fingers. Right, and so the big idea is, early Earth and origin of life, and major events in the history of life, mechanisms of macroevolution, and phylogeny in the tree of life. So petrosaurs, birds and bats, they don't have similar structure, right? So remember when I said that dinosaurs evolved into birds? Well, petrosaurs are not really considered dinosaurs; they're considered petrosaurs. But the dinosaurs evolved into birds, and so they had a different forelimb, a different style of forelimb. Where the bat came from mammals, so evolved in kind of mammals. So you see three different formations, but all the birds share the same style. All the bats share the same style, a little different, and petrosaurs share the same style. So when we're talking about homology, those are analogous features. Bird wings, bat wings, petrosaur wings are analogous. Same function, different ancestry. Homology may be different function, but same ancestry. Right? So early Earth formed about 4.6 billion years ago. Lots of good evidence to support all of that. Very difficult to scientifically deny the age of the Earth. As Earth cooled, and when it says the bombardment, we're talking about meteors and things like that. Those conditions were very, very different than what we see today. So there's probably a lot of water vapor and a lot of different types of gases, carbon dioxide, nitrogen, methane, ammonia, because there's a lot of volcanic eruptions going on, right? A lot of lightning, a lot of electricity in the air. Ultraviolet radiation was really, really intense. So our earliest evidence comes from stromatolites. And so those are 3.5 billion-year-old fossils. So that was kind of the first took about a billion years, which a billion years is a massive amount of time, and it's very difficult for most people to wrap their head around how much time that really is. But that's when we start getting our earliest evidence for life, those prokaryotic organisms, right? And the stromatolites are built by ancient photosynthetic prokaryotes, and they're still alive today. We can still see, not the same species, of course, they've speciated, but still same stromatolites, right? Because they use photosynthesis, it suggests that life first evolved a little earlier, perhaps 3.9 billion years ago, but our earliest evidence is 3.5 billion years. So here, if you look, what you're looking at here on the left are kind of iron rings, and when there's oxygen in the environment, iron will oxide, right? And so that kind of starts to show when oxygen was being pumped into the atmosphere. And then the big picture is kind of the early life, what it was like, the water, there's stromatolites, there's a lot of volcanic activity, things going on. So probably 4 stages, all right? So you have an abiotic synthesis of organic molecules, amino acids, nitrogenous bases, right? So we would have to show that those spontaneously can form spontaneously, right? If our assumptions are correct. And then joining these small molecules into polymers, such as proteins, right? Think of prions as just a protein cruising around, right? And then the packaging of these molecules into protocells. So we'd have to show, I think we showed earlier in what chapter when we talked about cells, that cell membranes would spontaneously form, right? And then the origin of self-replicating molecules that eventually made inheritance possible. So two scientists in the 1920s, A.I. Oparin and J.B.S. Haldane, they both proposed that organic molecules could have formed on the early Earth. And so our modern atmosphere's got a lot of O2, which oxidizes and disrupts chemical bonds. But when there's not a whole lot of O2, it doesn't disrupt chemical bonds as easily. And so the early Earth likely had a reducing atmosphere. So 1953, Stanley Miller tested the upper and Haldane hypothesis, and so he makes this apparatus. He puts gases in there and gives electrical charge, and he stimulates what the early Earth environment would look like, and then he set up a control, and boom, he made kind of nucleic acids. He started making amino acids. They spontaneously formed when we looked at the atmosphere and give it electric charge like lightning, right? So water vapor, methane, hydrogen, nitrogen, right? And then they looked at the analysis, and boom, stage one, abiotic synthesis, they found those organic molecules. He produced abundant amino acids, and so he tried other atmospheres and other energy sources, and he had similar results in all of them. So step one was demonstrated possible back in 1950s, right? And when we look at the number of amino acids that we now find from 1953 to 2008, we see the difference there. So other ideas about the origin of life: deep sea environments near submerged volcanoes or hydrothermal vents, right? They have a very unique life deep in the ocean there. Meteorites as a source of amino acids and other key organic molecules. And there's evidence for this, and when you open up meteorites, you can find the basic sources for amino acids, but it is more than likely the highest probability would be the Miller experiment. So stage two, the joining of monomers into polymers. So we have to take those basic units and we have to group them together. So when you have hot sand, clay, rock, any sort of heat, it can help that happens because it provides energy for chemical reactions. And when you have waves, bring those on to hot rocks, and then you can get polypeptides. This isn't really a stretch of the imagination. It can happen when you think of how big the Earth is and how much water there is and how many different locations. In fact, life, organic life arising from non-organic forms is not only possible, it's actually highly probable. Some people think that this is really, really improbable when you look at a lot of things. But if you look at a single instance, sure. But if you look at the billions and billions of instances in which this would be occurring every day across the Earth over a very long expansive time, then it becomes very probable. And so you can see kind of basic cell membranes, spontaneous form. We talked about that earlier. Packagings of polymers into protocells. So the sacs are vesicles. When you get lipids and mix them with water, we showed that these are spontaneously created, and then they're able to, these vesicles are able to grow and divide. And then now we need self-replicating molecules. And so we use DNA today, DNA to RNA, to protein assemble. So RNA molecules can assemble spontaneously from RNA monomers. So RNA can spontaneously assemble because of the laws of chemistry and physics, right? RNA monomers in the presence of RNA molecules form new RNA molecules complementary to the parts of the starting RNA. And then some, if you don't remember, the ribosomes are made up of RNA molecules, and so they can carry out enzymes like functions, right? So here's your collection of monomers, formation of short RNA polymers or simple genes, and then the assembly of the complementary chain. So that can show how life is highly probable, organic life is very probable to arise from non-organic life. So macroevolution, broad patterns of change of life on Earth. So we got 4.6 billion years of history. And so they kind of break it into lots of different, they break it into 3 eons and all sorts of different time frames to help us kind of sort it out. We have the Archean, the Proterozoic eons, and they put together, and then they lasted about 4 billion years, right, when you put those together. And then the Phanerozoic eon, that is the last half billion, so 4.6, right, 4, and then about 0.6. So when we look at prokaryotes and when they started and when oxygen started in billions of years, right, we see that there was a long time on the earth when there wasn't life, and then there's another really, really long period where you just got prokaryotes, a huge period where it's just very, very, very simplistic life, like the type of life you would find on Mars, right? And then we got to our single-celled eukaryotes and multicellular, and then just very recently we had this huge, huge burst. Land wasn't even colonized till the last half billion years, right? And animals were multicellular kind of animals, didn't arrive until about 0.7 billion years ago. So for a very, very long time, life was very, very simplistic, and it's only very recently that it's gone very complex. So yeah, prokaryotes lived alone on Earth for 1.5 billion years, from 3.5 to 2 billion years ago. So the prokaryotes transformed the atmosphere. Remember when I talked about iron oxide, and so that's the kind of physical evidence that shows that prokaryotic photosynthesis produced the oxygen, enriched the water in the atmosphere of the Earth. Anaerobic and aerobic cellular respiration allowed prokaryotes to flourish and move all around. And the oldest fossils of eukaryotic organisms, about 2.1 billion years. Common ancestor of all multicellular eukaryotes lived about 1.5 billion years ago. Oldest fossil multicellular eukaryotes are about 1.2 billion years ago. So single-celled eukaryotes, 2.1 billion, multicellular, 1.2 billion, right? That's a huge time frame. Then the first multicellular plants and fungi began to colonize land about 500 million years ago. So, like I said, when you look at 500 million years versus 4.6 billion years, it's really only very recently that we start to see this large diversity. Humans, we diverged from primates about 67 million years ago. Homo sapiens, or the Homo genus, originated about 200,000 years ago. And that's still adjusted as they find more transitional fossils, and they're still finding tons of transitional fossils for human evolution. It's really interesting. So this is kind of help you wrap your brain around the time frame. If Earth's history were compressed into an hour, humans appeared less than 0.2 seconds ago, right? So it just kind of lets you know when we're talking about 200,000 years ago, right? Then we think, oh, that's huge, a long time, but very, very recent. How do we know this? There's lots of different ways we know this. One of them, just one of them, is radiometric dating. So we're looking at the decay of isotopes. You should remember that from chemistry. So the rate of decay is a half-life, which is the time required for 50% of an isotope sample to decay, right? There's lots of different isotopes, right? We've heard of carbon-14. Carbon-14 only does a small time frame, but we have lots of other isotopes that we can do that they can handle much larger time frames. And so those half-lives range from thousands to hundreds of millions of years, right? So here's carbon-14. So when we talk about the fraction of carbon-14 remaining start at zero time, you can see this exact decay of half to quarter to eighth to 16th. It's very easy to see and very easy to graph and date. It's not, isotopes have a very, very predictable rate of decay. Additionally, you can use deductive reasoning, and you can look at the age of a fossil, infer the age from the rock layers above and below in which it's found. So the geologic record, and so you can do isotopes of the geologic record and make an inference about the fossils found in there. So when we look at geologic record for the most recent Phanerozoic eon, that's past 542 million years. We divide that into three eras: the Paleozoic, Mesozoic, and Cenozoic. And so the boundaries are marked by mass extinctions. So we've had a lot of mass extinctions going on. So remember that. So when we look at this part right here, right? This is 4 billion years that's going on, and so it is clearly not represented equally as this part up here. This part up here is only 500 million years. So we have to keep that in mind. However, we have the Cenozoic, Mesozoic, Paleozoic right here. Right? And this is when, first onset of animals, per se. And so we see lots of adaptive radiation during different extinction events. So first, we get, in the Cambrian explosion, they often call it, it's this huge increase in diversity. And there's some really, really freaky animal stuff that would be very sci-fi, and it's awesome when you start looking at some of those animal forms. And then you get a little marine algae abundant, and then you get colonization of land by diverse fungi, plants, animals. Then you get vascular plants. Devonian is the age of the fishes, first tetrapods, right? And insects appear. Carboniferous, we get these forests of vascular plants. First seed plants appear, right? And then reptiles and amphibians are dominant. And then the Permian, you get this huge radiation of reptiles, right? The origin of most present-day insects, and then you get this huge extinction of many marine and terrestrial organisms at the end of the Permian extinction. Then the Triassic, cone-bearing plants, gymnosperms, right? And then Jurassic dinosaurs start coming about, and Cretaceous, there's a lot more dinosaurs in the Cretaceous period than Jurassic period, but Jurassic is known kind of for that radiation. But we get our first flowering plants. First time flowers appeared, right? And then we start getting, there's a huge extinction at the end of the period again, and then we get a radiation of mammals. So after the dinosaurs died, it allowed niches for mammals. Angiosperm dominance, right? Those flowering plants kind of took over the vascular plants, so like the ferns and things like that. And then we get some primate groups, and then mammals sort of keep going, and then about we start seeing bipedal humans about 5.3 million years ago, and then our ice age in the 200,000 years ago, the first hominid appears. So, interestingly enough, they want to break this up into another epoch, and they want to call it the Anthropocene, because we humans have permanently altered the geologic record through our activities. So what that means is 10 million years from now, there will be this huge, there'll be an obvious change in the geologic record that can be attributed to human activity. So, macroevolution, how does this occur? Well, plate tectonics, right? This idea that the land masses can kind of move around, and if you look at it, it's got stitches like a baseball and different things like that. And so these plates kind of move around, float on underlying mantle, and so they call it continental drift. And then there's been kind of the idea of Pangea. There's been 3 occasions in which the land masses of Earth came together to form a supercontinent. So there's crust, there's mantle, outer core, inner core. And so that mantle is kind of viscous, right? It's fluid, it's lava, right? Things can move. So the landmass on top can move around. And here are some plates for tectonic activity. And when you put this around, it looks like kind of seams in a baseball. And so they call this the ring of fire up here, but you have plates all the way through here, right? I used to live in San Diego, and someone said, well, you're going to fall off in the sea, and that's not accurate. All it means is this whole, let me draw it in, well, that's not going to be a good color. It just means this big plate right here is turning this way. And it's just moving this way and spinning. It means in, several hundreds of millions of years, San Diego will be right up here next to Alaska. Right? So 250 million years ago, plate movements brought all the land masses together, and they call that Pangea. And during the Mesozoic area, Pangea started to break apart, which changes the environment and climate dramatically. Australia became isolated. That's why you get such unique life forms in Australia, right? And so here we see present, and so we see the difference, how Earth spread apart and how it was all kind of one landmass. So continental drift, right? The land masses moving can explain the distribution of lungfishes. It can explain the distribution of a lot of things. For example, near the Philippines, there's 2 islands really close together. In one you have the tree kangaroos, and the other you have primates. Right? And if they're so close together, why wouldn't the same mammals be there? Well, they weren't. The plates were moving, and so they were separated for a long time. And so it's just only recently that hundreds. For us, a really long time, but geologically, very recently, that those have kind of come together. So there's a lot of different things continental drift explains. So fossils of lungfishes are found on every continent except Antarctica, but living lungfishes are only found in South America, Africa, and Australia. So it suggests that lungfishes evolved when Pangea was still there. Here's a lungfish, they breathe there. So you see fossilized lungfishes everywhere in the living lungfishes in only certain areas. So volcanoes, earthquakes, they happen because the plates, the boundaries shift and move, and that movement is an earthquake. Undersea earthquake caused a 2004 tsunami when a fault in the Indian Ocean ruptured, and that caused that massive wave and caused all sorts of problems. So Pacific Plate, right? I used to live in San Diego. I'm very familiar with the San Andreas Fault, but this is the edge of the plate. This red line along here, this is the edge of the plate. Okay. And so this is what's spinning, this is what's moving, right? Remember the direction, this side is moving this way, okay? And then you have the North American plate, which on this side over here, this is a completely different plate. And so they're sliding across one another, and there's the San Andreas Fault. Extinction, right? We've had several mass extinctions. It's inevitable, yeah. 99% of all life on Earth has gone extinct. This says in each event, 50%. Some, I think, was it the Cambrian or the Permian extinction, I believe, where 90% of all Earth species went extinct. It was just incredible. Over the last 500 million years, 5 mass extinctions, and like I said, each time a ton of their species just went extinct. Imagine if it's on the low end, the 50%, just half of life as we know it just dies. Okay. Now, this extinction event, we have to understand the extinction events happen over a really, really long time, hundreds of thousands of years. Right? We're in the middle of another mass extinction event right now, but that's caused often by human activity, and it's higher than any rate of any mass extinction we've seen before. So an incredible extinction is occurring right now, but most of us don't see it because we only live for 80 years, 90 years, right? Permian occurred about 251 million years ago. And claimed 96%, I think I said 90%, 96%, and it was likely caused by volcanic eruptions. The Cretaceous caused all the dinosaurs except birds, and was probably caused by a large asteroid and caused global climate change, right? So here's your graphic, and they believe it hit in the Yucatan, near the Yucatan Peninsula, and if this is, if that is correct, you would expect to see some sort of crater, and in fact they've looked at it, and there is a crater that matches an asteroid impact right there, and it matches the physical evidence you would expect to see right there. So, every time there's a mass extinction, that leaves niches and allows for radiation. So it took 100 million years for the number of marine families to recover after the Permian mass extinction. 100 million years to get the biodiversity back. And the current extinction rate is 100 to 1,000 times the normal background rate, which means what we are doing today to our environment is going to take the Earth millions of years to recover. So good, bad, or indifferent, you have to decide that for yourself. But just be aware that human activity has a huge impact. So adaptive radiation. There's or periods of evolutionary change where you get kind of this extinction, and then you have a lot of unexploited areas of energy, right? And so you get all these new colonizations that kind of move out, right? Anytime there's competition for resources, if grass is greener on the other side of the hill, a lot of things run over the hill, right? And then they're in a new environment, so you get those adaptive radiations. So radiations may result from the evolution of new adaptations such as wings and petrosaurs. They can move around, birds, bats, insects. All of a sudden they can move to lots of different areas. And so adaptations for life on land in plants, insects, and tetrapods, right? So here's a reptilian ancestor and an ancestral mammal in one group. You get the dinosaur. Well, on one group over here, you get your monotremes, right? So your platypus, they don't have nipples. They kind of sweat their milk for their young, but they have hair, give milk. Even though they lay eggs, they're still mammals. And then you get another radiation of marsupials, right? And these are the ones that give birth really, really early and hold the young in the pouch. And then you have the urethran, or sometimes called the placental mammals, right? And so you see massive radiation. So here, right here is where the extinction of dinosaurs occurred. And so you see this rapid radiation going on after that. So if we look at the fossil records, and they can tell us when all these events occurred, and some really cool things, like what the great events were when they occurred. Continental drift, that's a big event. Mass extinctions, adaptive radiation, they all provide the big picture view of kind of how those things happen, how they come about. And so now we're able to understand these mechanisms that can describe the changes we see in the fossil record. We can understand that macroevolution. New one is evo-devo, evolution and development, right? The merger. A lot of evolution can occur in development. It's just kind of crazy. The genes that control morphological differences, can often just be one gene. We see lots of evidence where just one gene can control these huge differences. We also see that every group of animals has this similar setup. We have the same, they're called Hox genes. We have the same Hox genes as a fruit fly and a mouse. They're the exact same gene. But when they're turned on in us, it creates fingers. And they turn on in a mouse, it creates their digits. And they're turned on in fruit flies, it creates their legs. So it's the same gene, but a slight change can make a huge difference. So they can control the rate, timing, pattern, and change as it develops. So pedo, young, morph, so young morph, right? Pedomorphosis. So when we look at dramatic evolutionary transformations, when they can happen just because of the change in the rate or timing of development. So pedomorphosis is a retention of in the adult of body structures that were juvenile features in ancestral species. And so we see this in the salamander, which sexually matures but retains its gills and other larval features, right? So slight changes in relative growth of different body parts can change an adult form substantially. So just different changes at different times can create these huge difference in morphisms. So when we look at skulls of humans and chimpanzees, they're more similar as fetuses, but very different as adults, right? And so here's the human and chimpanzee fetus, but the adult is very different because the rate and growth, the development is very, very different. So homeotic genes, they're control genes. They determine basic features like where you're going to put wings or legs or whatever. And when those are changed, we have huge changes in body forms, right? We get and this is kind of showing mechanisms for that macro evolution. And then how or where they're expressed has profound alterations. So the duplication of development genes can also be important when the formation of new morphological features. So when we look at a fruit fly, it has a single cluster of homeotic genes. A mouse has 4 clusters of homeotic genes. And 2 duplications of these gene clusters occurred in the evolution of vertebrates from invertebrates, right? Those with backbones from those without backbones. The stickleback fish is an amazing example of Evo-Devo in action. When they live in the ocean, they have bony plates and spines to make it hard to eat, right, to help protect themselves. But when you put them in a lake, those spines, especially those pelvic spines, dragonfly larvae would use them to capture the fish and eat them. Dragonfly larvae are actually quite large, if you're unaware of that. And these fish are fairly small. And so we get freshwater lakes would have reduced or absent bony plates and spines. And it was a change in expression of just one gene. And it was just the expression. The gene was still there, but just when it was expressed, and we had this huge change. So here you see the pelvic spines, and here we see an absent pelvic spine, right? A complete morphological difference in fish, and it just would come from the control of a gene. The gene's still there because in order to express the jaw, they need that same gene. The same gene helps the development of the jaw, but the promoter region for the spines is absent. So in most cases, complex structures evolve by increments from simpler versions with the same basic functions. The evolution of an eye or other complex structure, behavior, or biochemical pathway, each step must bring a selective advantage to the organism, and it must increase the organism's fitness. So when we talk about the evolution of the eye, we see a lot of various steps, very simple steps, where we just see, kind of a pigmentation, and then it starts to get recessed, and so we see the eye evolve in complexity in lots of different ways. So we see kind of that transition where each step brought a selected advantage to the organism that had it. So mollusk eyes evolved from ancestral patch of photoreceptor cells, right? So just a patch of cells, and then they were adaptive. And so then we can see this whole range of complexity in mollusk. Cephalopod eyes are as complex as vertebrate eyes, but arose separately. They did not evolve because from a common ancestor, meaning they evolved in a separate lineage, which is very rare, but it shows how important the eye is. So here's kind of those transitional stages of a complex eye. Just patch of cells, and then you get a little cup, and then an optic nerve and fluid-filled cavity, and then we start getting lens, and then we get this complex eye that's very, very sensitive. In fact, if you go look at a squid or an octopus, they don't let you use flash photography. So really cool, we see this whole transition of the complexity of the eye evolve. So in other cases, some evolutionary novelties can result from the gradual adaptation of existing structures to new functions. So they take an existing structure and put it to a new function. And this is important. Irreducible complexity says that this can't happen, but this does happen. We see evolution co-opt different structures and then put together something with a whole new function. Such structures that evolve in one context but become co-opted for another function are also called exaptations, right? And they include feathers, because they're originally for thermal regulation, but they turned out to be pretty good for flight. Flippers, penguins, they were first for flight, but then they turned out to be pretty good for swimming, right? So the fossil record will reveal trends in the evolution of many species, but identifying these trends can be problematic. So when one example of this, when we look at horses, right? Because originally horses had a lot of different toes, and they've evolved to run on just one toe. They're kind of their middle finger, if you will. And occasionally, just occasionally, very rarely, you'll see kind of a horse with an extra little toe sticking out. Because in the development, because the genes are still there for multiple toes, they're just not expressed. So when we look at horses, if we select only certain species in the family tree, it appears that there was a general trend toward the reduction in the number of toes, larger size, and teeth modified for grazing. But if we look at all of the known members of this family tree, this apparent trend vanishes, right? So when we look at all the fossil record, so here's the ancestor right here, multiple toes, and here's the present-day horse with just one toe. And so we see grazers adapted for teeth and browsers, teeth adapted for browsing, but these guys all went extinct, right, through there. But we have to look at the whole picture. So branching evolution can lead to genuine trends, right? The species selection model of long-term trends compares species to individuals. So when they speciate, we consider it their birth, and extinction their death, right? So when it becomes a new species, that's when it starts, and then when they go extinct, that's when we consider it stopped right there. And then new species that come from them, we can kind of consider them offspring. This is leading you to phylogenetics, right? Unequal survival of species and unequal generation of new species play a role in macro evolution, similar to the role of unequal reproduction in micro evolution. So this is not, it's important to note that macro evolution does not occur by random chance. It's not random. It's the same deterministic laws that create macroevolution that are similar to the deterministic laws of microevolution. So evolutionary trends can also result directly from natural selection. For example, when we're looking at the horses again, when they invaded grassland that spread during the mid-Cenozoic, there was strong selection for grazers that could escape predators by running fast, right? So those are trends, and so we can this is a selective pressure. Predation is a huge selective pressures. Their two biggest selective pressures are predation and resource availability, right? So it's important to recognize an evolutionary trend does not imply that evolution is goal-directed. And in fact, evolution is not goal-directed at all. It's simply there's kind of this, I guess you could say, this downward escalator of natural selective forces, and evolution tries to keep up with adaptations, and animals generally don't make it. The average lifespan, like we talked about, is about 1 million years. They run on that, they're trying to run up that escalator for about 1 million years and eventually lose, and then there's open space for somebody else to, a new species to move in to that niche, right? So evolution is a result of interactions between organisms and the current environment, not interactions between organisms and what the environment might be. So phylogeny, right? It's the history of the species. It can be inferred from fossil record, morphological homologies, and molecular, right? We use a lot of DNA evidence for this. Homologies are similarities due to shared ancestries, and that's why we use DNA. So organisms that share similar morphologies are closely related, right? But Some similarities, right, are due to similar adaptations favored by common environments. So the shape of a shark and the shape of a dolphin, they're very, very similar, but they definitely don't have a common ancestor. But this is a process called convergent evolution because the environment is similar. They're both trying to swim around in the ocean a lot. A similarity due to convergent evolution is called an analogy. So these are analogous. You have the naked mole rat in Australia, right, and the North American mole rat here. And they're both very, very similar shape in the nose, right, curve here in the nose. Look at these big claws right here, big claws right here, short tail, kind of this really long body type. But one is a marsupial and one is a placental mammal. Okay. So systematics is a discipline that focuses on classifying, organizing, and more importantly, determining their evolutionary relationship. So Carl Linnaeus introduced taxonomy to name and classify. And he used the binomial, so that's genus and species, right? And that's kind of what most people are familiar with. And so then we go from there, we take the genera and we group them into progressively larger categories. And if you remember from chapter 1, do kings play chess on fine grained sand, right? That mnemonic to help you remember domains, kingdom, phylum, class, order, family, genus, species, right? So each taxonomic unit is a taxon. So here we go. Do kings play chess on fine grain sand? Okay. And so that's kind of how this works. So we are also animals. We're also chordates. We're also mammals, right? And so that shows our common commonality with the felines. So we are similar to the felines to class. Okay. Biologists use the phylogenetic trees to depict hypotheses about the evolutionary history and of the species, and so we use kind of this branching diagrams, and we call it a tree, but it doesn't necessarily end up looking like a tree, I guess. But these trees indicate the probable evolutionary relationships, the groups, patterns of descent, right? So here is a tree, right? So going from order, carnivora, right? We have Felidae, which led to the cats, and then we had the weasels and otters, and then coyotes and wolves, right? So from on order in the family, genus, and species, you can see kind of the relationships that break off. So the weasel and otters are more closely related to each other than they are to the cat, right? So cladistics was when we take and move things into what we call clades. And each clade has to be monophyletic, which is a group that includes all the ancestral species and all of its descendants. So let's go back here real quick. So this right here, we can call this a clade, right? And we can call this a clade, because this would be monophyletic. We can call this a clade, and that would be monophyletic. We could also call this a clade, and this would be a monophyletic clade, because at this point it has its descendant and all of its ancestor here and all of its descendants. But additionally, depending on what we want to do, we could call this right here, the whole thing, a clade, if we started here. So really what a clade is how we want to define it and what question are we asking, right? It depends on the question. Are we looking at relationships between weasels and otters? Or are we looking at relationships between the order Carnivora? It depends on the question as to which clade we would look at but each one has to be monophyletic, which means that I could not do something like, well, let's look at cats and dogs. Let's look at that group right there. We can't do that because there's a more recent common ancestor right here to weasels and otters, so we could not leave them out and call that monophyletic. So cladistics is based on the Darwinian concept that organisms share characteristics with their ancestors and differ from them. That's real important, differ from them. Thus, there are two main types of characters: shared ancestral characteristics and shared derived characteristics, right? Shared derived characteristics distinguish clades from the branching points in the tree. So an in-group is kind of what we want to look at when we're investigating whatever question we have, whatever, if we want to look at the relationships and cichlids, then that's our in-group. And then our out-group is some sort of, could be like a catfish or something like that. So we can have that out-group works as kind of a control to compare the relationship. So when we do the DNA studies and we look at all the cichlids, right? We have that catfish to compare it to because it's going to be very different than all of the cichlids put together, and that allows us to kind of build that tree. So outgroup, a taxon that diverged before the lineage leading to members of the group, right? And then we use this cladistics to identify the derived characters that define the branch and points of the in the phylogeny of the in-group. Right, so this one, guys, going to use a frog, and then tetrapods, and we're going to use presence or absence of traits. 1 if the trait is present, 0 the trait is not. So this is pretty simple here. Let's take a peek at it and see if I can explain it here. So our frog is our outgroup. So what trait? All of these are tetrapods, right? They each have 4 limbs, right? So what trait does the in-group here? So the in-group I'm going to put in blue. So here's our in-group, right? This is what we're looking at. How closely related is the duck-billed platypus to the iguana, or the kangaroo to that, or the beaver, right? The frog is the outgroup. We could include the frog, and then if we did that, then there'd be a fish right here, right? If we wanted to look at tetrapods, then we could use a fish as an outgroup, right? But in this case, the frog's the outgroup because we're looking for amnion, right, in the egg. This is very, very important. This allowed the establishment of animals on land permanently. A frog still has to go back to the water to breed because it needs that water to pull away the ammonia from processing proteins from the metabolism inside of the egg, where the lizard egg started using amnion, and so it could kind of do it itself and no longer needed water to do it. And so we can look at relationships there. And so from here we have hair, mammary glands, right? Like I said, platypus, they kind of sweat their milk, right? And then some sort of gestation, because platypus lay eggs just like the iguana. Kangaroos have a very short gestation, but they have a gestation, and then a long gestation, the placental mammals. And so over here we have the presence, absence of the group, of the character. So amnion, frog doesn't have it, doesn't have hair, doesn't have gestation, doesn't have long gestation, right? So zeros all the way through. The iguana has amnion, but doesn't have any of the other traits. So it's 1 or 0. Duck-billed platypus, amnion and hair, mammary glands. Kangaroo, hair, mammary glands, gestation. Beaver has all of these traits, right? So what they did is their tree is constructed from a series of branch points represented by the emergence of a lineage with a new set of derived traits. And so whenever they use phylogenetic trees, there's always a lot of conflicting, well, I don't want to say conflicting, a ton of information that's difficult to process. And so they always use parsimony, right? This is kind of like Occam's razor, looking for the simplest explanation for observed phenomena. So Occam's razor, the simplest explanation is probably the most correct. So when they do that, they use tons of different types of evidence, and when they do DNA studies, they look at a lot of different genes all over the place, and it gets very, very complex, and that's why there's whole entire computers, supercomputers, devoted to that, looking through a lot of this information. So the phylogenetic tree of reptiles shows that crocodilians are the closest living is very important, relatives of birds, because they share numerous features. They have a 4-chambered heart. We have a 4-chambered heart. Reptiles have a 3-chambered heart. Crocodiles have a 4-chambered heart. That's pretty cool. There's singing to defend territories, parental care of eggs. And so the traits were likely present in the common ancestor of birds, crocodiles, and dinosaurs. And we do see parental care in dinosaurs, right? So here's our outgroup, right? Lizards and snakes. Here's a crocodilian. And then how is it related to petrosaurs, ornithischian dinosaurs, saurischian dinosaurs, and birds, right? Of course, these guys went extinct. And so here are fossils of eggs and kind of protection and showing parental care in the dinosaurs. Using DNA, scientists have sequenced more than 110 billion bases of DNA, and you can go to DNA databases and tell it what gene you're looking for and what species, and you can build your own trees if you want, and it'll just, there's this huge database that's being contributed to every day. So if you want to go look up Homo sapiens and you want to look up a certain gene, you can look at all the A, G, C's, and T's that make up that gene, right? And so they use that, and here they are making a phylogenetic tree showing relationships of polar bears, brown bears, right? Very different than the black bear and Asian black bear, then your sun bear, your sloth, spectacle, pandas, raccoons, weasels, and red pandas, right? So even though this guy has the name Panda and this guy has the name Panda, they're not closely related at all. This guy's more closely related to weasels and raccoons, and this guy's more closely related to kind of these spectacle bears and sloth bears. So the more recently two species have branched from the common ancestor, the more similar their DNA sequences should be. That's kind of what they use, and it makes sense. The longer two species have been on separate evolutionary paths, the more their DNA should diverge. So you should see very different DNA from the red panda to the Asian black bear. But American black bear and Asian black bear should have more similar DNA than the polar bear and the brown bear. Right? And that's, of course, what we see. Longer twos, right, we talked about that. Different genes evolve at different rates. And so there's kind of different mutations in evolving. Sometimes we use mitochondrial DNA, or we use ribosomal DNA. because our mitochondria kind of has its own DNA. Remember the theory of endosymbiosis. Also, mitochondria only pass through the mother, so that kind of helps paternity all the way through. Ribosomal DNA has a slow change, and when we're looking at taxa that diverged hundreds of millions of years ago, we would look at ribosomal DNA. So one of the very interesting commonalities of molecular biology demonstrates that all living organisms share biochemical and developmental pathways, and a huge support of evolution, because if we came from a common ancestor, we should have a lot of these commonalities. And we do see that. And the ones that are most similar should have the most commonalities, and the ones that are least similar should have the least number of commonalities, and we see that. So the genomes of humans and chimpanzees are incredibly similar, and about 99% of the genes of humans and mice are detectably homologous. And about 50% of human genes are homologous with those of yeast. So we're not very closely related to yeast, but we're very closely related to chimps, and more so than we are mice, but we're closely related to mice as well. So molecular clocks, we can figure out when genes diverge because they have a reliable average rate of change. Okay? So it's a predictable rate of change, just like the isotopes, and so we can use that molecular clock and calibrate kind of how long things diverged. And we use this to date the origin of HIV infection, and so we kind of see when we look at this rate of change, we do the line of best fit, and so HIV kind of introduced from SIV, from the butchering of bush meat. So somebody with a cut on their arm was butchering bush meat, and some of the blood got on their arm or whatever. SIV mutated HIV and was introduced to humans about 1930-ish. Okay. So when we do this, we're constantly remodeling based on the number of data points. So just like science, it's always when we get better data or more data, it's willing to change. And so we currently recognize a 3-domain system. 2 domains of prokaryotes, bacteria and archaea, 1 domain of eukaryotes called, and that has plants, animals, and fungus. So molecular cellular evidence indicate that bacteria and archaea diverged very early in evolutionary history of life. And archaea are more closely related to eukaryotes than bacteria, which is very interesting, right? And so when we compare the genomes of all 3 domains, it shows that there's been substantial interchanges of genes between organisms in different domains, and it took place through horizontal gene transfer, and so that your plasmid exchange and viral infection, things like that. And some biologists like to suggest that the early history of life may be best represented by a ring from which 3 domains emerge. So it just kind of how you display it can vary, but the idea is the same. Recent common ancestors, things are more closely related. Common ancestors from further back, less closely related, right? But the endosymbiotic theory, right? You have gene transfer between mitochondrial ancestor and ancestor of eukaryotes. So, the eukaryotes creation, you have that kind of engulfing of that prokaryotic organism in that symbiotic relationship. Gene transfer between chloroplast ancestor and ancestor of green plants, right? And so they kind of see this ring, and here's eukarya and archaea and bacteria. I don't know if that helps you understand it, but There we go. That's our last chapter that we will be doing this semester. There is a 1409, which will start to go through each one of those branches, each one of those trees, and start to look at different things in more detail, which you start to really see the evolutionary relationships. That's it. Once again, there's no questions because the TechSmith software is not functioning. So just make sure you're keeping up on mastering biology and keeping up on the journals and discussions.